Good morning, students. Welcome back to Biology class, Chapter 9 Strategies for Enhancement in Food Production, Part 5. More than 840 million people in the world do not have adequate food to meet their daily food and nutritional requirements. A far greater number, 3 billion people, suffer from micronutrient, protein, and vitamin deficiencies or hidden hunger because they cannot afford to buy enough fruits, vegetables, legumes, fish, and meat. Diets lacking essential micronutrients, particularly iron, vitamin A, iodine, and zinc, increase the risk for disease, reduce lifespan, and reduce mental abilities. Okay, the plant breeding helping for the improved food quality. What are the advantages of the plant breeding? It is improving the food quality. Then the biofortification is the process of breeding crops with higher levels of vitamins and minerals or higher protein and healthier fats is the most practical means to improve public health. Breeding for improved nutritional quality is undertaken with the objectives of improving, first point, protein content and quality, oil content and quality, vitamin content, and micronutrient and mineral content. In 2000, maize hybrids that had twice the amount of the amino acids lysine and tryptophan compared to the existing maize hybrids were developed. Wheat variety Atlas 66 having a high protein content has been used as a donor for improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing over 5 times as much iron as in commonly consumed varieties. The Indian Agriculture Research Institute, New Delhi, has also released several vegetable crops that are rich in vitamins and minerals. Example, vitamin A enriched carrot, spinach, pumpkin. Vitamin C enriched bitter gourd, pathua, mustard, tomato. Iron and calcium enriched spinach and bathua. Protein enriched beans, broad, lab, French, and garden peas. These are the advantages of plant breeding. Plant breeding improved the food quality. Now next we'll study the single cell protein. From a single cell protein is synthesizing through the plant breeding technology. Conventional agricultural production of cereals, pulses, vegetables, fruits, etc. may not be able to meet the demand of food at the rate at which human and animal population is increasing. The shift from grain to meat diets also creates more demand for cereals as it takes 3 to 10 kg of grain to produce 1 kg of meat by animal farming. More than 25% human population is suffering from hunger and malnutrition. One of the alternate sources of proteins for animal and human nutrition is single cell protein. Okay, that is the alternate source of the proteins for animal and human nutrition is the single cell protein or the SCP. Okay, now microbes are being grown on an industrial scale as a source of good protein. So the one example, spirulina, is called a single cell protein. Why it is? Microbes like spirulina can be grown easily on materials like wastewater from potato processing, plants containing starch, straw, molasses, animal manure, and even sewage to produce large quantities and can serve as food rich in protein, minerals, fats, carbohydrate, and vitamins. Incidentally, such utilization also reduces environmental pollution. Okay, these are the important. Because of that, spirulina is called a single cell protein. Easily they can synthesize it. And also it contains all type of the protein, minerals, fat, carbohydrate and vitamins. Okay, and it reduces the environmental pollution. It has been calculated that a 250 kg cow produces only 200 gram of protein per day. In the same period, 250 gram of microorganisms like Methylophilus methylotrophus, Methylophilus, because of its high rate of biomass production and growth, can be expected to produce 25 tons of protein. Okay, instead of the 200 gram, 25 tons of protein we can synthesize from the microorganisms. The fact that mushrooms are eaten by many people and large scale mushroom culture is a growing industry makes it believable that microbes too would become acceptable as food. Next, we'll discuss about the tissue culture. As traditional breeding techniques fail to keep pace with the demand and to provide sufficiently fast and efficient systems for crop improvement, another technology called tissue culture got developed. Tissue culture was first developed by Haberland in 1902. The term Totipotent was coined by T.H. Morgan. And what does the tissue culture means? The whole plant could be regenerated from the explant. Explant means any part of the plant taken out and grown in a test tube under sterile conditions in special nutrient media. This capacity to generate a whole plant from any cell or explant is called totipotency. Okay. Now, the tissue culture is a technique of growing plant cells or tissues or organ in sterilized culture medium under controlled aseptic conditions. Okay, growing from a plant cell or a tissue or organ in a sterilized culture medium under controlled aseptic conditions. Then what are the steps involved in the tissue culture? First point, selection of the plant material. The part of the plant used for culture is known as explant. It is isolated from leaves, seeds, roots, buds, stem, etc. Then second process, preparation of the culture medium. A suitable culture medium is essential for the growth of its plant. The nutrient medium contains micronutrients, macronutrients, growth hormones like auxins, cytokinins, etc. And it is a carbon source material. Then the third process, inoculation. It is a process of transfer of explants onto the sterilized culture medium. Explants are transferring to the culture medium. Then the fourth point, callus formation. Callus means it is an undifferentiated mass of cells produced by the explant. We cannot identify root, stem or leaf, which part it is. It is an undifferentiated cells produced by the explant. Then fifth point, organogenesis. Organs are forming. What are the organs? Root, stem or leaf. It is a process of differentiation of cells in the callus into different tissues or organs. Okay, organogenesis. Then the next step, immediate exposure of the plantlets obtained from tissue culture to direct sunlight. Therefore, they are grown in a greenhouse for a brief period and then gradually transferred to their natural habitat. Okay, this is the last point. First it is in the bottles, then it is transferring to the natural habitat. Okay, these are the steps involved in the tissue culture. Here it is a explant, sterilization, totipotency. These are the points you should listen. Next it is an important to stress here that the nutrient medium must provide a carbon source such as source and organic salt, vitamin, amino acids, growth regulators, auxin, cytokinins, etc. By the application of these methods, it is possible to achieve propagation of a large number of plants in a very short period of time. The method of producing thousands of plants through the tissue culture is called micropropagation. Through the tissue culture, producing thousands of plants in a short period of time, it is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown. Okay, genetically and identically they are same from the parental plant, so they are called soma clones. Okay, soma clones. Many important plants like tomato, banana, apple, etc. have been produced on commercial scale using the micropropagation through the soma clones. Okay. Another important application of the method is the recovery of a healthy plant from diseased plants. Okay, application of tissue culture, micropropagation. Thousands of plants are produced in the genetically similar plants within a short period of time. Again, the healthy plants are producing from a diseased plant. The plant is infected with a virus, the meristem. Meristem means just growing region. The apical and the axillary. Apical means the tip region and the axillary in the side branches like. So there is free of viruses, just a growing region. They are not affected with the virus. So that region we can use for the tissue culture. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plants. Okay, which part is using for the virus-free plants? That is the apical meristem or the axillary meristem, because it is this area is the virus-free. Then next, scientists have succeeded in culturing meristems of banana, sugarcane, potato, etc. Okay. Scientists have been isolated single cells from plants, and after digesting their cell walls have been able to isolate naked protoplasts surrounded by the plasma membranes. Isolated protoplast from two different varieties of plants. What is the protoplast? The entire part. If it is a tomato, the entire tomato. Potato means the entire potato. Isolated protoplast from two different varieties of plants, each having a desirable character, can be fused to get a hybrid protoplast, which can be further grown to form a new plant. These hybrids are called somatic hybrids. The body cells are joining somatic hybrids, while the process is called somatic hybridization. Okay. The forming to the protoplast are hybrid fused to get a new one is called somatic hybrid. The process is called somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato, and then they are grown to form a new hybrid plant, that is the pomato, combining tomato and potato characteristics. But it is achieved, resulting in formation of tomato. Unfortunately, this plant did not have all the desired combination of characteristics for its commercial utilization. Okay, this is also another advantage of the tissue culture. First one, micropropagation. Second, it is the virus-free plants. Then it is the somatic hybridization. These all are the some of the importance of the tissue culture. Okay, thank you.